Good day, everyone. We're now at the top of the hour, ready to begin our webinar today titled Protein Engineered Nanomaterials. This webinar is being recorded, and you'll automatically get a link to the recording and to the slides. That typically comes to you a day or two after the close of today's live event. Our webinar today is brought to you by the Nanotechnology Coordinated Infrastructure, that's NCI, Southwest Center at Arizona State University, and the NAC Network Support Center at Penn State University, with grateful support from the National Science Foundation. This webinar is hosted by TeCentral. TeCentral acts as an information hub for the National Science Foundation's ATE grantee community. You can find out more about TeCentral by going to their website, as you see, tecentral.net, and about all the webinars that they host across the ATE spectrum, tecentral.net/webinars. Speaking of hosting, I'm your host and moderator today. My name is Mike Lasecki. I work with TeCentral and the NAC and NCI centers to help bring you this series. It's my pleasure today to begin by telling you about our presenters. I'd like to start by introducing Dr. Jin Kim Montclair, Associate Professor. She's the Director at the Center for Convergence for Innovation and Entrepreneurship. Rather, it's an institute, not a center, at New York University. Good morning or good afternoon, Jin. Welcome, and please introduce your two colleagues.
Hello. I'm really excited to be here. Today we'll have two of my researchers in the lab, Dr. Priya Kachel, who's a postdoctoral associate at New York University, and Lindsay Hill, who's an MD PhD candidate at New York University and SUNY Downstate Medical Center. So I'll begin. So today we'll be talking about protein engineered nanomaterials. And our talk is going to be divided into three sections. I'll be introducing the proteins as building blocks, which will then be followed by Lindsay, who will be talking about engineered protein fibers, which will then be followed by Dr. Priya Kachel, who will be talking about engineered protein nanoparticles. So today I'll be introducing you to proteins as building blocks for materials. So to take a step back, I'd like to talk about elements of nature. And through billions of years of evolution, nature has devised a means to create structurally diverse materials, shown on your left-hand side with different composite of different types of materials, or compilation. And these materials are able to produce or perform quite complex functions. For example, the leaf, shown in the top right corner, is able to harness energy from light. The spider is able to make this complex spider silk, which it is able to weave and be used as a home for the spider, as well as a mechanism to capture prey. And then below, to your left, is a magnetotactic bacteria that is able to crystallize these iron oxide nanocrystals, which then can allow this organism to sense the electromagnetic forces and allow it to swim accordingly. And then if you see on the right, bottom right, is a picture of hands, or if you just look down as the audience members down to your hands are an interesting source of materials in which, upon wounding, can self-heal. Now, all of these examples of materials or biomaterials are comprised of fundamental building blocks called proteins. And these materials, these materials are able to achieve complex functions through these proteins. So, proteins are comprised of 20 amino acid alphabet with defined order and number of secondary structural elements. And shown on the left are different types of proteins with different types of conformations. While our lab is interested in these proteins in nature, what we do is we take these structural elements that these proteins have, snip them out of their context and then piece them together in ways that nature has never devised. So we use molecular scissors and our molecular tape, and we hopefully build new types of materials with interesting materials properties. Now, on top of being able to do this cutting and pasting, we also are able to introduce non-canonical amino acids. That is, amino acids, which are the fundamental building blocks, but amino acids that nature has never thought of. So these are amino acids that you can't find in nature, that can be only synthetically produced. And what our lab does is we can introduce such types of non-canonical amino acids and combine them with these cut and paste proteins to produce new types of artificial proteins. So the way we produce our artificial proteins is we employ bacteria, and through advances in synthetic biology, we're able to achieve this by introducing genes of interest that contain the structural elements that we want, and then we, using recombinant technology, clone them into a host, a plasmid host, which then can be inserted into bacteria. And then our bacteria essentially serve as our factory to produce our artificial protein. Now, the artificial protein is then studied and explored, and our two speakers after me will be able to share with you some of the interesting materials properties and constructs that we've generated. So why are we interested in making materials out of proteins? Well, there's a certain advantage. So if we look at synthetic polymers, which are traditional materials that have been used, and in the left-hand side below, you have a synthetic polymer of polymethyl methacrylate, or PMMA. When you synthesize it, you get polydispersed materials. However, when you genetically encode proteins, you can gain access to monodispersed masses. And so an example here is of poly-L-glutamic acid, or PLGAs. And by defining the discrete genes of how many repeats we want, we can get monodispersed masses of our polymer. Moreover, we gain access to well-defined secondary structures, such as alpha helices and beta sheets. And so we can piece these defined secondary structures together, hopefully in ways that nature hasn't devised, and hopefully generate new types of materials with interesting material properties. So today, my researchers will be talking about constructing specifically smart biomaterials, and the types of materials will be shown as follows. So Lindsay will be talking about nanofibers and mesofibers, while Dr. Priya Kachal will be talking about lipoproteoplexes, nanoparticles, and hydrogels. And we make these materials not only just for their materials properties, but for a particular application. And our interest is for treating various disorders such as osteoarthritis, breast cancer, diabetic wound healing, diagnostic, and imaging. And so I'd like to introduce you to Lindsay Hill, who will be talking about engineered protein fibers.
All right, thank you, Jen, for that introduction. So I just want to start my part by noting that all of those examples that Jen showed from our lab included engineered mutants of this parent protein, the cartilage oligomeric matrix protein, or COMP as we call it. COMP is a protein that's a non-collagenous extracellular matrix protein. It's found in our cartilage, ligaments, tendons, and there it plays a role in the organization of the extracellular matrix. Now, COMP has 4 unique domain types, and the one of particular interest to our work is the coiled-coil domain, or COMP-CC, which I'm showing here. This domain is made up of 5 identical alpha helices, and that's a very common protein secondary structure. And these 5 helices wrap around each other to create this coiled structure that you see here. I'm also showing the primary amino acid sequence of COMP as it is synthesized in our lab. And what you'll see in red is that I've highlighted 6 histidine residues, and we call that the 6X his tag. This tag is able to chelate metal ions, and that's really helpful for us in purifying the protein in the lab. But as you'll see, chelating metal ions can also affect protein assembly, which is really interesting. Now, at the center of this coil are hydrophobic or relatively water-insoluble amino acids, and they create a hydrophobic pore, which has been shown by our lab and others to bind to small hydrophobic molecules, such as vitamins A and D, as well as anti-cancer drugs like curcumin and doxorubicin. And you can also see in blue a glutamine ring, and this ring of glutamine amino acids divides this pore into two. Now, additionally, this coiled-coil domain, or COMP-CC, can further assemble into nanofibers. These nanofibers have diameters of about 10 to 15 nanometers. And we can visualize them using a high-powered microscopy called transmission electron microscopy, or TEM, and that's what we're showing here on the right. And so early studies in our lab wanted to investigate which residues were really critical in COMP-CC's ability to self-assemble into the coiled coil and further form fibers. So here we're showing two single residue mutants of COMP-CC, T40A and L44A. T40A is a mutation at the 40th amino acid position from threonine to alanine, while L44A is a mutation from leucine to alanine at the 44th position. And as you can see in the TEM images, we found that T40A maintained the ability to form nanofibers, much like COMP-CC, but L44A eliminated fiber formation, meaning that that 44th leucine was really critical in COMP-CC's self-assembly. And we'll get into why in a moment. I first wanted to note that these mutants, when synthesized in the lab, also contain a 6X his tag. And that gives all of these proteins the ability to chelate metal ions. So our group was interested in investigating what impact chelating specifically zinc and nickel ions had on fiber assembly. We found out that these two metal ions actually had opposite effects on fiber assembly. Zinc bound to COMP-CC and T40A maintained fiber assembly and actually improved it. In contrast, it did not induce fiber formation in L44A. Nickel, on the other hand, initiated aggregation of the fibers, which you can largely see in the form of these clustered fiber maps. So two questions arose. Why did L44A eliminate fiber assembly, and why did these metal ions have opposite effects? To figure this out, we performed circular dichromosome spectroscopy, or CD. And this allows us to look at how proteins absorb circularly polarized light and see their their secondary structure. Different secondary structures result in different CD profiles. So in our case, we're interested in looking at the alpha helical content. And what you can see here on the right in green is that an alpha helical profile shows dips at 208 and 222. And then we can track if a metal or a chemical disrupts that alpha helical content by a loss of those dips until eventually we get a random structure, which we show in red here. So it turns out, which you can see in the solid black line, that a non-chelated COMP-CC and T40A show typical alpha helical structure. In contrast, L44A has a random structure. When they're bound to the zinc or chelated to zinc, all three proteins showed alpha helical content. In contrast, nickel, in all cases, disrupted the alpha helical structure, leading to a random structure for our proteins, which subsequently eliminated fiber assembly. So our lab wanted to then go a step further and mutate COMP-CC in an interesting way to engineer larger, more robust protein fibers. And that began with genetically engineering it to cut off the end portion, or the C-terminus, of our protein, as it was deemed unnecessary in in order to form our fibers and our coiled coil. So that C-terminus was eliminated, and then we performed what we call domain swapping, in which the two halves of the protein, a segment of amino acids prior to the glutamine ring and a segment further downstream of the glutamine ring, were actually swapped. And what this resulted in was a protein that we call Q. We call it Q as Q is the letter to symbolize glutamine, about which these protein mutations were based. And I just want to show here we have the primary sequence of the Q protein. Again, highlighted in red are our histidine amino acids. So that allows our Q protein to be further studied for metal chelation, much like we did with COMP-CC and the other COMP-CC mutants. But I think that the best way to really show the impact of these mutations on the protein Q is to look at a surface charge rendering of COMP-CC versus Q using protein modeling software. So here we're showing negative residues in red and positive residues are in blue. And what you can see is that while COMP-CC has a relatively well-distributed surface charge, Q actually maintains distinct patches of positive and negative charges. And under the correct pH, that allows for an electrostatic interaction between these positive and negative charges, resulting in both longitudinal and lateral fiber assembly to create a larger fiber formation. And so if we go back to looking at TEM images, we can see that compared to COMP-CC, which gave us fibers of about 10 to 15 nanometers in diameter, Q protein is actually able to form nanofibers with diameters from about 20 to almost 600 nanometers in diameter. So they really are much larger, more robust fibers. Now, we're interested in using these fibers as drug delivery vehicles, as biomedical applications are really important to our lab. So we wanted to take advantage of the protein's hydrophobic pore in order to carry small hydrophobic molecules, such as curcumin shown here, that would otherwise suffer in vivo due to their poor solubility in the aqueous or water-filled body. As I mentioned, a lot of these small molecules are hydrophobic, so they're not very compatible with our aqueous body. So for initial studies, we studied the protein's ability to bind curcumin, shown here. Curcumin is the active ingredient in the root turmeric, and it's a small hydrophobic molecule that's been shown to have antibacterial, anti-inflammatory, and anti-cancer properties. But as I mentioned, its in vivo use is limited due to its hydrophobicity. So we hope that by carrying curcumin within the protein's hydrophobic pore, we can actually aid in its delivery to the body, such as to cancer cells. Curcumin is also a great molecule for proof of concept or early studies, as it's trackable based on its absorbance at 420 nanometers, as well as its fluorescence at 530 nanometers when it's bound to the protein's hydrophobic pore. So when it's bound, you can actually visualize fluorescent fibers using fluorescence microscopy, which I'll show in just a bit. So compared to the TEM image where we had nanoscale fibers, after binding Q protein to curcumin, we're now getting micron scale fibers. So that's 1,000-fold bigger. And the image on the right is taken from a fluorescence microscope, and it shows drug-bound fibers, where you see that now we have that 1,000-fold increase in fiber diameter. And that's really great for visualizing our fibers. And we're currently using these drug-bound fibers in our lab for in vitro cancer cell studies. So we think they're really promising as drug delivery vehicles. Now, as Dr. Montclair had mentioned, we can incorporate non-canonical or non-natural amino acids into our protein. So on the left, I'm showing one of the naturally occurring amino acids, leucine. And on the right, I am showing a non-natural or non-canonical variant of leucine called trifluoroleucine. And as the name indicates, it's much like leucine, except that 3 hydrogen atoms are replaced with 3 fluorine atoms. Now, fluorine is known to stabilize proteins against high temperatures as well as chemical disruption. But you can also use fluorine for fluorine MRI. Now, there's limited fluorine in the body, so any signal that you get from fluorine MRI is going to be very specific to your fluorine-incorporated construct. So here I'm showing a traditional proton-based MRI at the top, and at the bottom we have a fluorine MRI image overlaid onto that proton MRI, and you can see in red and orange, that's your fluorinated construct, and its intensity is very specific to the location at which that fluorinated construct is injected. So our lab wanted to utilize trifluoroleucine in our Q protein. So we synthesized a trifluoroleucine incorporated Q, and it was found that these large fibers had increased thermostability as well as increased chemical stability. We furthermore wanted to study how they interacted with the zinc and nickel ions, much like we did with COMP-CC and its single residue mutants. So it turned out that much like with the previous mutants, zinc further stabilized the fibers. In contrast, nickel chelation disrupted fiber formation and instead yield aggregation, which is similar to what we had seen before. So our lab is currently using these trifluoroleucine incorporated Q fibers, not only to further optimize them for chemical and thermostability as well as drug binding, but also for fluorine MRI applications. So with that, Mike, I'd like to ask if there are any questions from the audience. I'd be happy to take them.
Lindsay, thank you. There is a question, and it's clear from your presentation that you've been able to engineer these fibers from small nanoscale features right up to that what you refer to as a mesostructure of a couple of microns or more. Is there a target? I mean, would you like to make a 1 millimeter fiber, or you're customizing the size of the fiber for the application, such as drug binding? I mean, what are you doing when you think about size? Just making them bigger,
yeah,
or what do you do?
I think that depending on the size, these fibers have different applications. Staying on the nanoscale may better allow for vascular injection. In contrast, the mesoscale allows for an amplified amount of drug binding and more of a scaffold-like material for direct injection. So I think that it really has to do with the application that you're looking for, whether you want a drug-filled scaffold or something that can be injected throughout the vasculature. I think this mesoscale fiber formation was really novel, and it was interesting to us that we could go further than nature and make these mesoscale fibers in contrast to the nanoscale fibers that exist. So yeah, it's really application dependent, but I think that it seems like we can tailor it based on our genetic engineering and drug binding, tailor it to what we're interested in.
It's good. It's interesting work. I have one other question. I'd like to invite Dr. Montclair to come back on. Dr. Montclair, you responded to a question in the chat about self-assembly. When these proteins assemble, do you guide them? Is it a natural thing that you just accept the result? I'm not sure I'm asking that question the right way, but a lot of us here in the nanotechnology world think about self-assembly, and I was wondering how that applies in this case.
Right. Great question. So our proteins are designed to come together. So we had engineered them based on the original natural existing protein called cartilage oligomeric matrix protein, coiled-coil. And that actual structure assembles into these, 5 of them come together into these pentamers that can self-assemble. And so naturally they have a hydrophobic region that allows the self-assembly to occur, and then the outside surface is water soluble, so that so that it can do this in under aqueous environment. So it's programmed into the actual coiled sequence that allows these proteins to self-assemble in that way. Now, what Lindsay had shown was that we can then take this protein and re-engineer it by swapping some domains so that now on the surface we have patches of positive and negative charge, and it's that positive and negative charge that allows it to further assemble or self-assemble into thicker fibers, going from a nanoscaled fiber to more mesoscaled structure.
Very interesting. There's another question that has come in, and it's from an admitted non-biologist. They say, well, gee, when we see those scissors and talk about scissors and tape, it's not always clear how that actually happens. I mean, you know, what does scissors mean in this context? How do you actually make that cut? Is it physical? Is it chemical? Right. How does that actually happen?
Right. So it's a very good question. And yeah, so it's relying on a lot of biological principles. And so the scissors that I'm referring to are usually either enzymes or restriction enzymes that can cut nucleic acids. So nucleic acids are what we're using, or the genes that encode the proteins. We're developing these genes, and then we're inserting them into a host so that we can produce the protein, so that those genes get translated into proteins. And so when I'm talking about molecular scissors, I'm actually talking about doing some genetic engineering in which we're using enzymes, restriction enzymes that can cut DNA. And then we have molecular paste or glue, and those are what I refer to them are actually ligases. So these are enzymes that can actually ligate DNA or put together two pieces of DNA so that it seamlessly becomes one strand, one actually gene. And so we have scissors that are used in the form of restriction enzymes, and then tape in the form of ligases in order to do our genetic engineering to clone the gene that we want so that we get the protein that we want. Does that make sense?
It does. It does. It's all chemistry. Well, maybe not strictly, but anyhow. Okay, good. Thank you very much, Dr. Montclair, for that response, and Lindsay, for your presentation. Colleagues on the line, make sure that you continue to put your questions in the chat window. We're going to go ahead into our next section right now. Just a moment while I adjust the system here. One moment, please. There we go. So Priya, welcome. Dr. Patel, welcome, and tell us about engineered protein nanoparticles.
All right. Thanks a lot, Mike. So changing gears here a bit, though still building upon the concepts discussed by Lindsay and Professor Montclair, I'll talk about protein-engineered nanoparticles. I'll give you 3 different examples of these nanoparticles and its related applications. Now for the first example, I'll talk about our protein, which is COMCC supercharged protein, or CSP in short. Now in order to make a supercharged variant of COMCC, we change few of the residues in the protein sequence to arginine, which is a positively charged residue indicated by R in this lower sequence, which is the bottommost sequence that you see. Now arginine residues being on the surface of the protein renders the surface charge of CSP as positive, which is shown in blue color in your right panel. So the more blue color it is, the more highly charged it is, the more highly positive charge it is. Now, being a positively charged protein, it can readily bind to negatively charged nucleic acids like DNA and RNA. And here we are showing a representative example of a double-stranded siRNA, which are short interfering RNA that interferes with the expression of the specific gene and its associated protein. Now, this protein siRNA complex packs together to form another complex called as packs together to form a proteoplex, which is a protein and the siRNA complex. And what we do next is we use these liposomes or lipid vesicles to further encapsulate this proteoplex particle. So taken together, the protein, the siRNA, and the lipid forms another complex or a larger unit called as lipoproteoplexes. Now, the advantage of using our lipoproteoplexes is that these assemblies protect the nucleic acid from degradation and allow it to readily pass through the cell membrane. And henceforth, these formulations can be used for gene delivery. And these assemblies, or this lipoproteoplex, or you can also call them lipid protein complexes, these assemblies are of about 160 nanometers in size as shown by transmission electron microscopy, which is the figure that is at the very bottom that you can see. Now, as Professor Montclair and Lindsay mentioned that we are interested in looking for the biomedical application of these formulations, I'm going to give you an example of one of such applications. Now, to assess the efficacy of these lipoproteoplexes, wounds on diabetic mice were treated with a formulation containing the specific RNA, siRNA, that knocks down the expression of a protein which is upregulated in diabetes. And the wounds were monitored until the wounded area, so we use the highlights in here. So this is the wounded area, and it was monitored until till it resembles the surrounding intact skin, which is right here. I'm going to close this down. So with our lipoproteoplexes, which is the lower panel, it took about 22 days for the wounds to heal as compared to 31 days in untreated mice, which is the top panel. So overall, for this example, we demonstrate that our formulation, like our lipoproteoplexes, can be used for gene delivery, and more so, it can enhance the rate of diabetic wound healing and lessens the wound burden. So coming to the next one, so the next two examples are based on COM-CC block polymers. So by block, we mean that we are using domains of different or same protein and arranging in different fashions. So for the first block, we are using COM-CC or C, which is right here, which comprise of a coiled-coil self-assembly having a hydrophobic pore. And to this hydrophobic pore, various small molecules can bind. And for that example, actually, Lindsay talked about curcumin delivery by Q fibers. So curcumin binds to the hydrophobic pore of this COM-CC assembly. And in terms of structural elements, this assembly comprises of alpha helices, so this ribbon-like structure that you're looking at. For another block, we included a different protein called as elastin, or we refer as E, or it can be also referred as elastin-like polypeptide. Now, E exists as random covalent solution, and upon a change in temperature, it collapses and assembles and self-assembles into beta spiral, something like this. Elastin is composed of a pentapeptide repeat, which is VPGXG, which is like the sequence is right here. So X here is an interchangeable amino acid. The properties of E, more specifically the mechanical properties of E, can be tuned by varying the X amino acid and the number of repeats. Now what we did next was right here. So we combined both C and the E to form another protein called as CE, and since it has one block of C and another block of E, you can also refer to as a diblock. So and the whole new construct is known as CE. Now in this example, we employed CE during this hexahistidine tag, which is like 6 histidine residues in red in the sequence, for the templated synthesis of gold nanoparticles to yield this beautifully self-assembled protein gold nanoparticle assemblies, or we can refer them as nanocomposites. And these assemblies are of about 23 nanometers in size as depicted by transmission electron microscopy. Now, as you know from Lindsay's presentation, that curcumin, curcumin is insoluble in aqueous conditions and doesn't penetrate the cells, we investigated whether our protein gold nanocomposites could enhance small molecule delivery, more specifically for curcumin, and uptake by specific cells called as MCF7 breast cancer cells. Now, both CE, which is protein by itself, and CE bound to the gold nanoparticles, complex with curcumin, indicated by the CCM, exhibited uptake as visualized by the fluorescence microscopy. So the left panel, which you see right here, is just with the protein itself bound to curcumin, and the right one is CE bound to gold nanoparticles and complex with curcumin. And the curcumin appear to be present within the cytoplasm of these cells. Now, this was further confirmed by quantifying curcumin extracted from these cells at 420 nanometers. Extraction of curcumin revealed twofold greater amount of available curcumin for CE gold nanoparticles, which is shown in this red bar right here, relative to the protein by itself, which is shown in black. So here we give you an example that curcumin bound to CE was taken up by the cells and with gold nanoparticles enhancing its uptake. Coming to a last example. So here we are showing you a tri-block protein, where we added this additional C domain to the CE protein that we discussed. And since it has 3 blocks, which is COM, elastin, and another COM, and that's how we refer it to as CEC. So basically it has E domain flanked by 2 C domains. Now this protein self-assembles into nanoparticles, which we also refer as micelles, of about 50 nanometers in size. Now, dynamic association of these particles leads to hydrogel formation, which you see as the thin protein sheet on the right. Now, just to give, I want to specify something here, is that the image on the left, which is the nanoparticles, is visualized under transmission electron microscope, while the image on the right, which is the hydrogel, is the result of micelles packing together to form this protein sheet. And this is visualized without any aid of a microscope, and hence it's like at a macro scale. And currently, we are studying these hydrogels for its various applications, including drug delivery and biomimicry. So in closing, or as summary, I would like to say that using COM-CC, we prepared a diverse set of nanomaterials, including fibers and nanoparticles. And we are currently studying these smart biomaterials for different applications, ranging from drug delivery in osteoarthritis, gene delivery in diabetic wound models, and development of fluorine-based theranostic agents. So if someone has any questions regarding these, we'll open the floor soon. And lastly, before I hand it over to Mike, I want to thank the audience, and special thanks to Bob and Mike for having us and giving us this opportunity. And Mike, do we have any questions from the audience?
We do, Priya, and thank you very much for your presentation. I'll look at a question that came into the chat window that Dr. Montclair has started to respond. Let me take you back 2 slides, Priya. I'm taking you backwards right now. The question had to do is, you showed some results in cell cultures, for example, the tissue healing in mice. how far, in a general sense, is this technology away from beginning clinical trials? I mean, where people are using, bioengineered, bio-inspired protein fibers in actual clinical situations. Are we 15 years away? Are we 3 years away? What would you think?
Based on our result, it seems promising, I would say. So I can't say the exact numbers because still like a lot of optimization needs to be done, and then mouse is just one case, and before it goes to the actual human trials, a lot of optimization in terms of different ratios and the optimized dose and frequency of these formulations needs to be done. So I would say still maybe 5 years or so. I'm being optimistic here. Or maybe Jen, let me could answer.
Well, let me.
Go ahead, Jen.
Yeah, so I think it's more down the line, closer to 15 years. The reason why I think so is because we also have to get FDA approval for these materials, and we have not done that at all. And so that process takes quite a bit of time before we can do any type of clinical types of trials. So most clinical trials are done with FDA-approved materials. And so since these are materials that have not been studied before except in our lab, we would have to go through that process. I would imagine that would.
Sure, that makes sense, and thank you. It's just that people get excited, right? They know how diabetic wound healing is a big issue, and of course breast cancer issues, and they see promising ideas like this, and they start wondering how long till application. But I understand the whole sequence of the trials. So thank you for that response. Priya, let me try this question on you. You can see it in the chat window. Does the increase in curcumin uptake, is it due to confirmation with the gold nanoparticle, or just the increase in the number of molecules?
So I'll go back to that slide actually. This is right here. So what we believe is maybe this is a better example. So if we have protein by itself, it does exhibit uptake. And yes, it's true that once it comes together, it forms a large assembly, and it has more number of COM, and we believe that curcumin binds to the hydrophobic pore of the COM. So the more number of COM it has, the higher the concentration of curcumin can be bound to it. So I believe, yes, somewhat kind of both, and I do believe the increase in number of molecules is enhancing the uptake. And more so, yeah, I believe so, unless, I don't know if Lindsay or Jen have anything to add.
Lindsay, do you have a comment there?
I'm looking, I'm not sure about this one so much, but I was looking at the next one that's also related to the uptake of curcumin. I did have a comment on that one. So one of the next question was asking about curcumin. Let me see here. Well, I can just use this. If having curcumin mixed with the gold would also help in this uptake. And I think that they're playing two separate roles. The issue with that would be that curcumin in and of itself is still going to be a hydrophobic molecule and have low bioavailability. So I think that it really does benefit from having a drug carrier. And the gold in and of itself may not lend to improving that bioavailability so much as something like the protein, which has a hydrophobic pore. I think that that's really what's going to help in aiding the delivery of these small molecules rather than the gold alone. Sure,
that makes sense. I'll rephrase this question and ask Dr. Montclair if she might have a response. If you think about, as you look at research like this and you realize that some of these are natural molecules like curcumin, is there any evidence that you're aware of that, ingestion or potentially, adding your own gold, so to speak? I know that's not an engineered gold complex, but are you aware of anything physiological effects that might benefit humans from doing these things. I don't mean to put you on the spot, but there might be some interesting adjuvant type of research there.
Right. So in terms of gold, I'm not sure, but certainly with curcumin, yeah, people in Asia use curcumin for food, and there's some evidence for lower susceptibilities to cancers as in terms of preventing, but I'm not sure about healing. So if you have cancer, if you ingest curcumin, I'm not sure if that'll completely heal the cancer. So there is some evidence of that in terms of just ingesting it in food. So having some curcumin in your food is good for your health. With gold, I'm a little less knowledgeable in terms of whether ingesting gold would be helpful. There's also toxic side effects of any type of metal that you're going to be ingesting. So I'm a little more cautious about prescribing or just suggesting that gold should be used, and people should start ingesting that.
Sure. Well, good. Well, thanks. I mean, I just want, it's a logical question, of have there been other similar studies that we might learn from in this way. So thank you for that response. Priya and Lindsay and Jen, this slide, this application slide really strikes me because today we've seen all sorts of applications, even fluorine magnetic resonance imaging, with these larger fibers. That was very unique. And how you've been able to really structure not only the appearance of these fibers, but their functionality, their dimensions, using these approaches. And I think, your approach toward bio-inspired really makes sense here. I'm going to take us forward as we wrap up our presentation today. So hold on just one second as I gain control here. Here we go. And in addition to your thanking us, we would like to thank you for your presentation today. And we really enjoyed the work that you, Dr. Montclair, and your colleagues are doing at New York University. So thank you for the time. Friends, as we get near the end of our webinar today, I'm going to put a survey on the screen. It'll open in a separate browser window, and it's just 4 brief questions. We'd like you to respond to it. And presenters, please don't respond to the survey because sometimes it accidentally closes it, but we'll just leave that alone. So I'm going to launch that now, and as that survey comes up, I'll make our final presentation, presentation of thanks to our presenters. So hold on, folks, just one moment while I click the survey links. Now, for every computer might not work in exactly the same way, but that survey should open in a separate browser window. If it doesn't, we'll send that survey link back out to you at the conclusion of this webinar. Remember, the webinar has been recorded. You will get a link to the recording and the slides. So in closing, I'd like to thank our colleagues from New York University, Dr. Montclair, Priya, and Lindsay for their excellent presentation today. So thank you. Folks, that officially ends our webinar. Thank you for joining today. Presenters, you can just go ahead and log out or go on to mute as we now end the recording of our session. Thank you again.
Thank you.